Hello dear students, welcome to the class. Today we are going to learn about photosynthetic pigments. Okay, let's begin. So you can read on the screen that I have already given notes on that to save our time. See, what is the meaning of pigments? Pigments are the substances which absorb light at a particular wavelength. Okay, for example, if I talk about chlorophyll A, right? This is one pigment, right? Which does photosynthesis. So many types of chlorophyll are there. I'm talking about chlorophyll A, which is the main pigment for doing photosynthesis. Chlorophyll A can absorb sunlight at many wavelengths, right? But it will show maximum photosynthesis at which wavelength? Maximum photosynthesis will be done at either 680 nanometer or at 700 nanometer. Right? So can I say that 680 to 700 nanometer maximum photosynthesis occurs? This is a specific wavelength or not? Right? This is a specific wavelength. Right? So pigments are substances which are absorbing light of a particular wavelength and they are doing photosynthesis. Understood? Fine. Now, how we understood that different types of pigments are there? You know that today we have chlorophyll A, chlorophyll B, xanthophylls, carotenoids, all these different types of pigments we have learned in this chapter in NCERT, right? So how we understood about all these different types of pigments? So pigments are separated by the help of a technique which is called chromatography. Specifically, there are different types of chromatography, but specifically paper chromatography is that technique by which we understood how many different types of pigments are there. And chemistry also, you have learned that chromatography is one technique by which if there are so many things present in a mixture, you can separate the you can say components of that or constituents of that mixture, right? So let's suppose for this what we have to do, we have to take leaf and we have to make leaf extract with the help of mortar and pestle. Okay, after crushing the leaf in mortar and pestle, we will make leaf extract, and then that leaf extract we have to put a drop with the help of a dropper on this chromatographic paper. Okay, what is this thing? Chromatographic paper. Okay, you don't have to learn the entire technique, but at least you should know that how we understood about different types of pigments. So in this paper chromatography, there is two phases which are there. One is called mobile phase, okay, which is liquid, and another one is this one, which is paper. This is called stationary phase. Okay, what happens here? As soon as with the help of a dropper you have dropped your leaf extract over here in the paper, what will happen? As soon as this paper will be touching the mobile phase, which is actually liquid, right? This mobile phase will be starting to get absorbed by the paper and it will move up, right? As it will touch this particular drop, it will start taking the particular components or constituents from this drop and it will segregate as per the as per the mobile phase is moving in the stationary phase. So you will see some bands or colors in this paper, right? Like for example, the band which you are going to see will be blue color band, okay? Like this, a blue color band. Okay, then maybe green color band. Okay, then yellow color band. Okay, and then you will be having orange, yellow to yellow orange color band. So you have got these different bands here, right? How you got it? As the mobile phase was moving, all the components, all the particular components of that mixture, that leaf extract got separated, right? So different colors bands were observed here, which were actually different pigments, right? So total how many pigments we have found with the help of this paper chromatography? We have obtained 4 different types of pigment: chlorophyll A, chlorophyll B, xanthophylls, and carotene. Now you have to be very careful. You have to remember chlorophyll A was appearing of which color in this particular band? Chlorophyll A was appearing to be bright blue Okay, bright blue, green in chromatogram, right? This was the color of chlorophyll A. Chlorophyll B was having which color? It was having green color, yellow green color. It was having yellow green color. Xanthophyll was having yellow, right? And carotene, yellow to yellow orange. Okay, these are the colors which you have to remember, because in this particular data they can ask you questions like match the following. They will give you on one side they will give you these different pigments, on another side they will give you the different colors. So they can ask match the following, odd one out, incorrect pair. These kind of questions are possible here, so this is important to remember. Now how you will remember this, right? To remember this, there is a trick which you can follow. Okay, what is the trick? You know about this term, right? With cure. Okay, now what you have to do, you don't have to consider violet, you don't have to consider indigo. Okay, this blue, bright blue to green, what which color you have written here? Bright blue green color is for chlorophyll A. So bright blue and green for chlorophyll A, right? Then for chlorophyll B it is yellow green, so yellow green for chlorophyll B. Okay, only yellow for what? For xanthophyll. Okay, and yellow to yellow orange. Okay, yellow to yellow orange is for carotene. Okay, you don't have to consider, right? Okay, so by this also you can remember. Okay, maybe this trick will help you. Now we have to study about these different pigments in detail. So please remember that this particular chlorophyll A, okay, this acts like a chief photosynthetic pigment or the main reaction center pigment. Okay, maybe out of all these four pigments, if you have chlorophyll B, xanthophyll and carotene, but you don't have chlorophyll A, then photosynthesis is never going to happen, right? So chlorophyll A is the main chief photosynthetic pigment without which photosynthesis cannot occur, right? Then it is universal photosynthetic pigment. Each and every organism, whether it is algae, whether it is bryophyte, whether it is pterido, gymno, angio, whether it is cyanobacteria or any particular bacteria which is doing, you can say photosynthesis, they must have this green pigment which is called chlorophyll A. This is a universally occurring photosynthetic pigment. Each and every photosynthetic organism must have chlorophyll A, right? Then this actually is present in the reaction center. Okay, I will tell you later what's the meaning of reaction center. In reaction center, the main pigment which is present is one single molecule of chlorophyll A. Okay, one single molecule of chlorophyll A forms reaction center. So this you have to remember. Okay, now what about the other pigments like chlorophyll B? We have learned xanthophyll and carotenes, these three. Okay, one more confusion is there. Xanthophyll and carotenes together they are called carotenoids. Okay, xanthophyll and carotene together they are called carotenoids. So if they have written xanthophyll and carotene, it's fine. They are talking about yellow to yellow orange type of pigments which are carotenoids. Okay, anyways, chlorophyll B, xanthophyll and the carotenes, hundreds of these molecules make a structure which is called accessory pigments. Okay, and these accessory pigments together they make a structure called antenna complex or light harvesting complex or shield pigments. So what you have understood? You have total four pigments, right, in leaf: chlorophyll A, chlorophyll B, xanthophyll, carotenes, right? Chlorophyll A is the main reaction center pigment, right? Single molecule of chlorophyll A makes main reaction center, and it is universally occurring in each and every photosynthetic organism. Then rest three which we have got, these three, they make a particular complex which is called light harvesting complex. Okay, and this light harvesting complex, it is also called antenna complex, shield pigments, and these pigments are also called accessory pigments because they help in photosynthesis. They actually don't do photosynthesis directly. They help the main reaction center pigment, right? Main reaction will be done by chlorophyll A. These three help it to do so, right? Now I will tell you how these four pigments are organized. These four pigments are organized in the form of a particular unit called photosystem. Okay, what is photosystem? It is a photosynthetic unit. Where it is present? It is present on the thylakoid membrane, right? The pigments, which includes the main reaction center pigment. What is the main reaction center pigment? Chlorophyll A. And the accessory pigments. What are the accessory pigments? What are the shield pigments? What are the light harvesting pigments? They are chlorophyll B, xanthophylls, carotenes. They are present on the thylakoid membrane. Not randomly, they are organized in a particular system which is called photosystem, in a particular unit which is called photosystem. So these pigments are organized into photosynthetic unit called photosystem, and these photosystems actually are present on thylakoid. This is what I have written. Now I will show you how a photosystem looks like. So by now you understood that this is our thylakoid membrane, right? On this thylakoid membrane, this is how the photosystem appears like. Okay, a photosystem is actually a multi, you can say, pigment proteins complex in which lot of things are present. These things which are drawn here with yellow color, you can consider they are accessory pigments. Okay, some of them can be chlorophyll B, some can be carotene, some can be xanthophylls. So these are accessory pigments. And here in the center, you have chlorophyll A, which is the main reaction center pigment, right? Then you have certain more things like you have primary electron acceptor, like this. Okay, certain enzymes are also associated here sometimes. Okay, these things makes a photosystem. Okay, so you understood the photosystem. You have accessory pigments, you have main reaction center pigment. Okay, what is this chlorophyll A? This is main reaction center pigment. Main reaction center pigment. Okay, what is this primary electron acceptor? What are these things? Enzymes. Certain enzymes are associated sometimes, right? So these four things make a photosystem. So understood, pigments are not here and they are spread across the thylakoid membrane. They are organized in a way, in a manner, in a unit on the thylakoid membrane in a form of photosystem, right? Now you have two types of photosystem. Okay, two types of photosystem are there. And this depends on the main reaction center pigment. Okay, so based on the main reaction center pigment, you have two types of photosystem, photosystem 2 and photosystem 1, right? Photosystem 2 and photosystem 1 are named based on the time of their discovery and not in the order in which they are located on thylakoid membrane. So please remember, first discovered photosystem was photosystem 1. That's why it was named as PS1 or photosystem 1. Then later we understood that one more photosystem is there, so we named it as photosystem 2. But on the thylakoid membrane, what is located first? It is photosystem 2, then we have photosystem 1, right? So this PS2, PS1, this is the name given based on the discovery time, not based on where they are located. Okay, now you have these characters and these are the photosystems which you have to remember. First point, just read one by one. Main reaction center pigment in photosystem 2 is chlorophyll A, which is showing maximum absorption at 680 nanometer specific wavelength, right? Photosystem 1 is having chlorophyll A, which is showing you absorption at 700 nanometer. Okay, so one is showing maximum absorption at 680 nanometer, that is photosystem 2. Another photosystem is showing maximum absorption at 700 nanometer. Then main reaction center chlorophyll A showing maximum absorption at different wavelengths in both the photosystem. That's why we have two types. Then light harvesting complex, same in both, right? It is made up of chlorophyll B, xanthophylls, carotene, and all these all these pigments are bounded to protein, right? Tell me how many chlorophyll A molecules must be there in one reaction center? 2, 1, or more than 2. Okay, then primary electron acceptor is different in both. In NCERT, primary electron acceptor is not given for photosystem 2, but I have written here. Pheophytin is the name of primary electron acceptor of photosystem 2, and ferredoxin, which is mentioned in NCERT, it is the name of primary electron acceptor of photosystem 1. You have to learn all this. Okay, any particular enzyme is mentioned for PS2 in NCERT? No. But one enzyme is given in NCERT which is associated with PS1, that is ferredoxin NADP reductase, FNR, ferredoxin NADP reductase. And what is the location where you are going to find photosystem 2? Grana. Grana is that part, remember, the stacks or piles of coin, that part of thylakoid, it is having PS2. right? PS1 is present both on grana as well as stroma lamellae. Remember stroma lamellae, that particular thylakoid which is connecting to grana. So PS1 is present both on grana and stroma lamellae, but PS2 is exclusive to grana only. Okay, understood. Now, if accessory pigments are not acting like main reaction center, then what is their role in photosynthesis, right? So we have different roles for accessory pigments. You can see I have written here. Accessory pigments are actually enabling more wavelengths to be used up during the photosynthesis, right? Okay. Can anyone tell me what is the meaning of photosynthetically active radiation? Hmm? Almost 400 to 700 nanometer, right? This is the range of wavelength in which sunlight, like this is the range in which the light can be used for photosynthesis, right? But I am telling you that 680 nanometer is the maximum absorption of chlorophyll A of PS2 and 700 nanometer maximum absorption of chlorophyll A of PS1. Then what about the other wavelength, which ranges from 400 to 700 nanometer, which is not absorbed mostly by this. So accessory pigments, they are going to utilize, they are going to absorb that wavelength, and then by resonance energy transfer, they will transfer the energy after absorption to the main reaction center pigment. Because of this, what happens? Lot of energy together is not falling on chlorophyll A, right? Because as a result, what will happen? If all the wavelengths are absorbed by the same pigment, it will get damaged, right? That will result into photo-oxidation, right? That much energy it cannot hold because all the electrons will be lost at once and the structure will be destroyed. So what we have done, what plants have done, they have segregated the roles, right? They have separated the absorption. Chlorophyll A is only absorbing sunlight of 680 nanometer, 700 nanometer, that wavelength, and other wavelengths like 400, 450, 500, 550, 600, these all wavelengths are absorbed by different pigments, maybe chlorophyll B, maybe xanthophyll, maybe carotene. And after absorbing, it is giving the energy to main reaction center pigment, right? So that is the reason that carotenoids are enabling wider wavelength of incoming light to be used in the photosynthesis. Another thing, by doing so, they are helping in maintaining the integrity and the structure of chlorophyll A and avoiding the problem of photo-oxidation, right? That's why they are called shield pigments, shielding the chlorophyll A, because that's main reaction center pigment. Understood? Okay. Now, very quickly we will read about these graphs also. We have two graphs here, absorption spectrum and action spectrum, which are given in NCERT, and then they have clubbed both the graphs and given one more graph. So I will be taking these two graphs. You can see in absorption spectrum what they are telling you. This is a graph which is showing you the absorption of different pigment at a particular wavelength, right? So it is telling you the degree and the portion of wavelength absorbed by different pigment, right? This is the meaning of this graph. Now what you can see that here, this blue graph is for chlorophyll A. Chlorophyll A is showing maximum absorption in red region and blue region, right? So this side blue region, this side red region. Okay, maximum peak, just notice. Okay, chlorophyll B also you can see somewhat similar only, right? And then you can see this yellow graph is for carotenoids. So what you are looking here, you are looking that at which wavelength, how much absorption which pigment is showing. This is the meaning of absorption spectrum graph. And you can compare them also, right? Can you see chlorophyll B is showing more absorption in blue region as compared to chlorophyll A, right? Because the peak here is more than this, right? And on the same time, at the same time, you can see chlorophyll A is showing more absorption in red region as compared to chlorophyll B, right? So this is the meaning of absorption spectrum. Action spectrum is showing you that how much photosynthesis is occurring at a particular wavelength. Okay, so it represents the effectiveness of different wavelengths of light to perform photosynthesis. So you can see that both in the red region, right? You can see in the red region and blue region only maximum photosynthesis is being done, right? Then if you will merge these two graphs together, you will get to know that action and absorption spectrum gives information that maximum absorption occurs by pigment chlorophyll A in blue region and red region. Okay, and if you understood this, okay, this is a line which you have to learn. After that, you can see I have given one question. Graph absorption spectrum depicts that chlorophyll A shows maximum absorption in both blue and red region as compared to chlorophyll B. You just try to analyze absorption spectrum graph and give the answer of this question, that can you say that chlorophyll A is showing maximum absorption in blue region also and in red region also as compared to chlorophyll B? Just now I have told you. Okay, just see in both the region chlorophyll A, is it showing maximum absorption as compared to chlorophyll B? Just give me the answer of that, right? So this was all for today. Thank you everyone for watching the video and all the best.